Welcome. My name is Ellen Crocker, and I'm a postdoctoral scholar with the Forest Health Research and Education Center at the University of Kentucky. Thanks for joining us for the first of our video research seminar series.
Today I'm joined by Dr. Anna Conrad. She'll be talking about her research. Dr. Conrad's area of expertise is in how trees respond to biotic and abiotic stress. She also looks at chemical-based screening tools and their role in tree improvement programs. Thanks, Dr. Conrad, for coming.
Thanks, Ellen, for that nice introduction. So today I'll be talking about the role of phytochemicals in tree responses to abiotic and biotic stress. So, as many of you are probably well aware, trees are long-lived organisms that have the ability to live in a variety of different environmental conditions. This is truly a testament to their resilience and that ability to not only respond to, but also defend against these various environmental conditions, which can include abiotic and biotic stressors. So, the outline of my presentation today will be phytochemicals and tree stress response. What exactly do I mean by phytochemicals? Then I'll talk about two different projects that we're working on at the Forest Health Research and Education Center that seek to study this relationship. The first is on evaluating phytochemicals as markers for hybrid chestnut disease resistance to Cryphonectria parasitica, the causal agent of chestnut blight. This is a more applied project. And the second I'll be discussing results of a project that we're working on, looking at the association between phytochemicals and dormancy in two different perennial tree species, peach and apricot. So first, what are phytochemicals and how are they associated with tree stress responses? So phytochemicals can be simply defined as plant-produced chemicals. They're found not only in all plant species, but also all tree species. They can be further categorized into two primary groups, primary metabolites and secondary or specialized metabolites. Some common examples of primary metabolites found in trees and plants include ethanol and also the amino acids like the amino acid phenylalanine. Some common examples of secondary metabolites include terpenes or terpenoids. These are compounds that are often found in pine and are what resin is made up of. In addition, flavonoids, like the compound quercetin, is common in many different tree species, especially in hardwood tree species such as oak. So the rest of my presentation is going to focus on plant secondary metabolites, or PSMs. PSMs in particular are known for their association with tree stress responses, and this includes both abiotic stresses such as drought or cold temperatures, and biotic stresses such as plant pests and pathogens. So the types and amounts of PSMs are tree species dependent. This means that different tree species will contain a different suite of PSMs, and even within a tree species, these compounds may differ, or the concentrations of these compounds may differ. In addition, PSMs can be temporally and spatially variable. And what I mean by that is that different tree tissues, such as the bark, the leaves, or the roots, may contain different compounds or different concentrations of the same compounds. And they may change depending on what time of year it is. A good example of this is differences that we see in the color of leaves in the fall foliage, as displayed at the bottom of the screen. As you can see, different trees have different color leaves in the fall, and this is because of different chemical composition of those leaves. For example, we can contribute the green coloration of leaves due to high levels of chlorophyll, whereas yellow and brown colors are associated with high concentrations of carotenoids, and red and purple colored leaves are associated with compounds called anthocyanins. So what's the role of PSMs in tree stress response? So these compounds are structurally and functionally diverse, and as a result, they can play many different roles within the tree. For example, they can provide protection against abiotic stressors. So some PSMs are known to protect trees against drought and the accumulation of reactive oxygen species, which occurs when trees become drought stressed. And also, some PSMs, such as flavonoids, can act as tree sunscreen to protect trees against UV light, much like sunscreen protects us, our skin, against the sunlight as well. In addition, many of these compounds can provide protection against biotic stressors, such as pathogens and pests. And there's multiple different ways in which these compounds may protect trees. For example, PSMs may be directly toxic to pathogens and pests. They may contribute to the formation of lignin, which is a structural barrier within tree cells. They may also indirectly protect trees against pathogens and pests by serving as signals within the tree to let the tree know, other tree parts know that the tree is under attack. And finally, some of these compounds may serve as pro-oxidant or antioxidant activity, protecting trees against these stressors. Finally, these compounds can be produced before, constitutively, or after induced stress. So next I'm going to be talking specifically about one project we're working on at the Forest Health Research and Education Center, where we are evaluating phytochemicals as markers for hybrid chestnut disease resistance to Cryphonectria parasitica, the causal agent of chestnut blight. First, let me talk about the role of chemicals as markers for resistance. So, while some chemicals may play direct roles in tree resistance or defense responses, they may also just be associated with that response. So it's a correlation, not necessarily a causation. So the goal of this type of work is to develop models that predict tree susceptibility based on the tree's chemical composition. So the question that we ask can be displayed by the picture at the bottom of the screen. Can we measure all the chemicals or a subset of the chemicals present within a tree, and will that allow us to predict susceptibility of a tree to a given pathogen or pest? So one way that we go about addressing this is through an approach called chemical fingerprinting. Chemical fingerprinting is a comprehensive analysis of all the chemicals present within a given sample. This can be a tree tissue like leaves or bark, or it could be an extract from those same tissues. In this approach, the individual chemicals are not separated, identified, or measured. Instead, we're looking at a snapshot of the entire chemical composition of a given tissue at a given time. Since this may change depending on the tree tissue that we're interested in or the time of year, it's important to take that into consideration when using this approach. So one way that we collect chemical fingerprints and how we're doing it in chestnut is we're using a technique called Fourier transform infrared or FTIR spectroscopy. So using an FTIR spectrometer, such as the one pictured here, we are able to collect chemical fingerprint data from many different chestnut hybrid samples. So an example of a representative chemical fingerprint or chemical spectrum collected using FTIR spectroscopy is also pictured on this screen. So each chestnut tree will contain a unique or have a unique chemical fingerprint or chemical spectrum. So we can use that data to ask the question of whether or not chemical fingerprinting can be used to screen hybrid chestnut for disease resistance prior to infection with C. parasitica. So C. parasitica is a devastating pathogen that causes the disease chestnut blight. And symptoms of this disease that are characteristic include canker on the stem of the tree, as you can see in the picture at the bottom of the screen. So if we combine our chemical fingerprinting data with knowledge of the level of susceptibility of a given tree based on the symptoms such as canker lesion length or a rating that can be assigned, we're able to determine whether or not this approach may be useful for screening trees for disease resistance. So what we found in our research is that in certain cases, in certain groups of trees, we are able to use chemical fingerprinting to predict the level of tree susceptibility to chestnut blight. Specifically, we've identified certain regions within that chemical fingerprint or that chemical spectrum that we use to do this, to predict the lesion length. Now, because we only tested this in a relatively small set of trees, we need to perform additional evaluation to determine how reliable and robust these models are at predicting chestnut hybrid disease susceptibility. However, regardless, there is potential for more high throughput analysis in the field or forest setting using handheld instruments such as the one that's displayed at the bottom of the screen. This would greatly facilitate current efforts aimed at breeding resistant chestnut hybrids and reintroducing chestnut to our eastern forests. So, finally, I'm going to talk about another project that we're working on at the Forest Health Research and Education Center, where we're studying the association between phytochemicals and dormancy in two perennial tree species, peach and apricot. Now, phenology is a study of cyclic events and the influence of climatic and seasonal variability on these events. So we can view phenological traits as adaptations of trees to abiotic stress, such as cold temperature and lack of water. So some common examples of adaptive phenological traits in trees include winter dormancy, bloom date, which is the date in which flowers appear on trees, and chilling requirement. So some trees require more or less chill in order to flower and then eventually fruit. So the objective of this study is to examine the association between adaptive trait variation, such as the examples I listed before, dormancy, bloom date, and chill requirement, and the production of certain phenylpropanoid intermediates. Now, phenylpropanoids are one group of plant secondary metabolites, and we're examining this in two different tree species during dormancy. Now, we've focused on 8 chemicals that that others had found to be associated with dormancy and also seed stratification in herbaceous plant species. And 3 of those compounds include the flavonoids apigen, camphorol, and quercetin. In order to evaluate this, we collected floral buds from peach and apricot trees that varied in chilling requirement throughout endodormancy and ecodormancy. Those are 2 phases in dormancy. The first being where some factor within the tree prevents the tree from growing, that's endodormancy, and ecodormancy is when the environmental conditions of that tree are preventing it from growing. So we looked at the variation in the concentration of those phenylpropanoid intermediates in high and low chill peach and early and late flowering apricot. Based on past research, we knew that variation in the chilling requirement was associated with genetic differences in these trees as well. So what we found is that in high chill requirement peach and late flowering apricot, the concentration of flavonoids, including quercetin, camphorol, and apigen, spike in buds prior to blooming. And then those concentrations drop just as the buds start to bloom, or where we see the appearance of petals. This time point roughly corresponds to the endodormancy-ecodormancy transition in trees that require greater chill. However, we also found that this pattern was less uniform in low chill requirement peach and early flowering apricot. So why do we care about the changes in flavonoid concentration that we observed during dormancy, and also differences in those concentrations between trees that were differentially adapted to cold temperatures, the high and low chill peach and the early and late flowering apricot? Well, flavonoid aglycones, such as those that we measured in this study, are known negative regulators of auxin transport. Now, auxin is a plant hormone that plays a critical role in plant growth and development. In addition, flavonoids can also scavenge reactive oxygen species, which are produced as a byproduct of auxin catabolism. So, flavonoids, in fact, may be playing an important role in tree growth and development. However, the question that comes from this research is what exactly is triggering the changes in flavonoid concentration? We know that there are differences, and we know that these differences are associated with differences in the trees. However, what environmental conditions are triggering it? Is it changes in light intensity, which occur during dormancy, changes in temperature, or perhaps changes in the availability of water? So as dormancy progresses and temperatures warm, the ground becomes less frozen and more water is available for trees. So which of these factors may be impacting the flavonoid concentration? We don't know, but hopefully future research will be able to address this question. So some take-home messages from today's presentation. Phytochemicals play important roles in tree responses to abiotic and biotic stress. They can be used as markers for disease resistance in trees in certain cases, which would have important implications for tree breeding programs where a major limitation currently is time and spatial resources. In addition, from our work studying the relationship between phytochemical concentration in peach and apricot during dormancy in trees that had adaptive trait variability, we determined and we concluded that phytochemicals do have a role in regulation of, or likely have a role in regulation of plant growth and development. So before signing off, I'd like to acknowledge those who've collaborated with me in working on these different projects and our funding sources. Thank you.
Thanks so much, Anna. That was really interesting. Thanks for teaching us about phytochemicals as well as your research. I feel like there's some great basic biology in there, but also really important applications to breeding American chestnuts that are resistant to chestnut blight and better trees in general. We really appreciate it.
Thanks so much, Ellen.
If you want to learn more, please visit our website, foresthealthcenter.org, and hope you join us again for another research seminar.